Bismillahirrahmanirrahim. My name is Salat Naz, and today we will have a lecture on monocyte macrophage cell system, which is basically associated with white blood cells. And this system is also famous as reticuloendothelial system. So the objective of our lecture is to quick review of the types and functions of WBCs, factors responsible for the start of phagocytosis, and discuss about the process of phagocytosis, neutrophils and its role in defense system of the body, monocyte macrophage cell system. White blood cells are also known as leukocytes, and basically they are divided into granulocytes and agranulocytes. The granulocytes are basically neutrophils, eosinophils and basophils, and agranulocytes are lymphocytes, monocytes and macrophages. They are the mobile unit of the body's protective system. So basically the neutrophils and neutrophils, eosinophils, basophils and monocytes. These four collectively, they play a role in the phagocytosis. Whereas the lymphocytes, they secrete antibodies and their role is basically in the immunity. Function of white blood cells. As you know, they are the fighting forces in the human body. These are like we have army, we have navy, we have air force. Similarly, in our body, we have different types of forces like neutrophils, eosinophils, basophils, monocytes, and lymphocytes. The neutrophils they give defense against the bacteria and fungus. Eosinophil give defense against the parasitic infections. Basophil give the responsible for the allergic responses. Monocytes are basically give rise to another type of cell which are macrophages, and that play a role in the phagocytosis of bacteria or foreign agents or any chemical or the dead cells. And then we have lymphocytes. They provide defense and immune system. But before we move towards our main topic, reticuloendothelial system, I would like to discuss three more main important factors that are basically play a role in the reticuloendothelial system. First one is the chemotaxis. It is the movement of the cells, means defense cells towards the chemical substances. And these defense cells are basically the neutrophils and macrophages, which move towards a chemical substance, and these chemicals are basically chemotactic substances, that is they attract the neutrophils and macrophages. And what are these tactic substances? These are bacteria or viral toxins and degenerative products of inflamed tissues, because whenever there is inflammation there is some degeneration and there is release of different substances. And the product of complement complex activated in inflamed tissue. Whenever there is some inflammation, there is complement complex is formed, and this will serve as the chemotactic substance. As well as the byproduct produced by plasma clotting in the inflamed area. You know, whenever there is some inflammation, there is plasma clotting outside the cell. So the byproducts of this is also serve as the chemotactic substance for the cells, defense cells. Now other important process is the diapedesis. It is a process by which neutrophils and monocytes, you know, only the blood cells have neutrophils and monocytes, and then we don't have the macrophages at this stage. So these neutrophils and monocytes, they follow a pathway where the pores will open, and there is a squeezing of these cells, because you know the neutrophils and monocytes are large in size. However, the pores inside the blood vessel are small. So what will happen? These cells will squeeze, you know, they are round. Okay, now they are squeeze, सिकुड़ गए हैं बीच से, ठीक है, थोड़ी अपनी जगह बना के वो they will move outside the cell, and this movement is known as diapedesis. And then there is chemotaxis, that is, this is the bacteria which is attracting this neutrophil. Okay, so we have discussed about the chemotaxis and diapedesis. Diapedesis is basically a special type of movement through which the neutrophils and monocytes will leak out of the blood vessel so that they can come towards their target substance. And then we have another term which is very common. We have already know about this amoeboid movement. This is a special type of movement by which the neutrophils and macrophages they move towards the damaged area, because neutrophils and the macrophages are the cells which engulf the bacteria. Means they will take inside the bacteria into their body, you can say, for the body of the cell, you can say. So begin with the protrusion of one end of the cell. You see, this is the protrusion, means this is the enlargement at one part of the you can say the macrophage. Okay, and then this will move. For example, you can see in the picture that these are the bacteria. Okay, now this whole cell has to move over there. So first of all, they will create some false foot, they say. We also call it pseudopodium, and these pseudopodium will then interrupt these bacterial cells. So all the rest of the cell will move to, for example, this will also give rise to another pseudopodium, and all it will move towards this direction, and it will engulf these bacteria. Okay, so question arises, why normal tissues are prevented from the phagocytosis? Why the white blood cells do not eat our own cells? Sometimes this happens. This is an abnormal condition, but normally it does not happen, because phagocytosis is a process that ingests the wrong agent. Okay, so the phagocyte, which is the cell including monocyte and macrophage. So the phagocyte is selective, means selective to what? Rough surface, protective protein coating, opsonin. What are these? We will discuss here. So, the most natural structures in the tissues have smooth surfaces which resist phagocytosis, means do not allow the eating to itself, smooth surfaces. So, the substance to be phagocytosed has a rough surface. If the surface is rough, then it gives chance for the phagocytes to come and engulf that particular cell. It may be dead cell, it may be bacterial live cell, or so on. So rough surface is very important for to become the selection of the phagocytes. And then we have a protective protein coating. Most natural substances of the body have a protective protein coats that repel the phagocytes. So most dead tissues and foreign particles have no protective coating. So basically, rough surface and protective protein coating, these are the factors that are important for the selection of the phagocytes. Another factor is the opsonin. Opsonin is basically antibodies. Antibodies, you can see, adhere to the bacteria. If you can see, this is the antibody that is attached with this bacteria, and this antibody is not from the body, means it is a foreign body for us. So what will happen? Presence of this antibody make it susceptible for phagocytosis. This is the macrophage, and it is attracted by these antibodies that this is not the part of our body, and we have to remove this from our body. So macrophage will move, and it will eat the whole bacteria inside it, and then there's a process of degradation and so on. Phagocytosis basically consists of three steps: recognition and attachment, engulfment, killing, and degradation. So this is the whole story over there, how these phagocytes work. to remove the pathogens or kill the pathogens. So basically neutrophils has the ability, they are small cells, so they can kill 3 to 20 bacteria, small size bacteria, and macrophages are very large in size, so they can kill 100 bacteria, they can engulf 100 bacteria and kill them. So phagocytosis of the pathogens such as bacteria by phagocytic cell, this is the phagocytic cell such as macrophage, and this figure is about the macrophage. So the antibodies, you know, in the as I already tell you in the previous slide, that these are the antibodies which are the source of selection. So the antibodies coat the bacteria, making them more susceptible to phagocytosis by the macrophage, and this engulf the bacterium, bring it into the cell and forming a phagosome. Now what will happen? Because of this op, op, because of this these antibodies, which are the foreign bodies, and it is detected by the phagocytes. So what will happen? There is a formation of pseudopods. Okay, pseudopods will move towards this microbe, and they form the phagosome first of all. Okay, they engulf it and form the phagosome, and phagosome plus lysosome, they form phagolysosome. Phagolysosome perform the function of digestion of microbe, and then there is a process of exocytosis, and microbial debris will be moved outside this cell. So this is the whole process of phagocytosis, both in the neutrophil as well as in the macrophages. This is a huge macrophage again, and you know the macrophage is too much large in size that it can engulf the neutrophils. It can even engulf the red blood cells. So they are very large in size, and they can kill the bacteria, and you know, they can engulf about 100 bacteria at a time. They are so much large. Oh my god. So now come, this is another macrophage, and again this is showing the same thing, that there is a microbe, and the chemotaxis cause the movement of this macrophage towards its microbe, and there is a pseudopodia movement, and then there is adherence of the microbes, and these microbes are in contact of phagosome, and then there is a formation of phagosome, then phagosome with the in the presence of lysosomes, they combine, and then they form phagolysosome, because there is lysosomes, killing of microbes by the enzymes and other chemicals occur at this stage, and then there is some residual bodies or residual particles that will eliminate by the process of exocytosis. Role of neutrophils. Up till now we have discussed about how phagocytosis occur. From now onward we will discuss about which cells are involved in this process and how they participate. So neutrophils form an essential part of innate immune system, and recruited to the site of injury within minutes following the trauma. It is a hallmark of acute inflammation, and neutrophils undergo chemotaxis. Cell surface receptors allow the neutrophil to detect the chemical. Role of neutrophils have 3 stages for direct attacking the microorganisms. So basically the neutrophils work in 3 ways. First is the phagocytosis, that is ingestion of the bacteria. Then release of sometimes it is not possible for the neutrophils to ingest the bacteria because of the large size. So at least what they will do, they will release soluble antimicrobials, including granule proteins that will act on the bacteria, so that at least they can control their function. So and the other option is the generation of neutrophil extracellular traps by the neutrophils. So phagocytes cytosis, release of soluble antimicrobials, and the neutrophil extracellular traps, these are the main ways of targets. Now you see, these are, this is the bacteria, and this is the traps. This is the web like structure. So the activation of neutrophils causes the release of web like structure of DNA. These neutrophil extracellular traps, these are the extracellular traps, comprise a web of fibers trap and kill the microbes extracellularly. This picture show you how the neutrophils are working. First of all, they are in the circulation. Then once they get the signal that there is some inflammation or the foreign particles enter into the body, so they will move towards the edges and then stick to the edges of the vessel wall, and then there is a process of diapedesis that we already discussed, so it will move outside the blood vessel, and then there is a process of chemotaxis. With the help of chemotaxis, they will attract towards a foreign body, but they will take some time and recognition and see if it is a foreign body or not. So there is opsonization, that is, they will see there is antibodies or not, so it will detect, and then if there is opsonin present over there, what they will do, they will ingest this material, this bacteria into it, and then there is a degranulation, that is release of chemical substances that will degrade this bacteria, and there is oxidative killing, and finally netosis occur, that is there is some release of these substances, as well as there is formation of network, so that they will stop the movement, or you can say delay the inflammatory process. So, and this picture, this is from the Guyton. This picture shows you that these are the neutrophils. Okay, these are the neutrophils which are already attached with these endothelial cells. But when the cytokines, these are the cytokines which are basically released from the inflamed area. When these cytokines reach over there, okay, they will remove the neutrophils from this attachment, and the neutrophils will perform diapedesis and move out and move into the tissues. Okay, so this is how the neutrophils are moving outside and then act on their target cell. Now, monocytes. Monocytes are, of course, another type of cells. Monocytes have two main functions in the immune system. First of all, they replenish, or you can say they fill up the resident macrophages. For example, macrophages has been used during an anti-inflammation process. So we need more and more macrophages. So this is done by monocytes. So, and the second function is in response to inflammatory signals, monocytes can move quickly, and it take about 8 to 12 hours to reach the site of infection in the tissues and divide and differentiate into macrophages and dendritic cells to elicit an immune response. So monocytes basically are the provider of macrophages, so that macrophages will engulf the bacteria and provide immunity or defense against the cells. So this picture show you that initially these cells are pro-monocytes, which are then when they come into the blood, they are converted to monocytes, and when they come into the tissues, they become macrophages. Monocyte macrophage system. This is a complete system in the body. So the monocytes, they become macrophage after entering the tissues. Initially, monocytes are in the blood, and once they reaches the tissues, they become macrophages, and once they reach the tissues, they become attached to that particular tissue. So every tissue has some pool of macrophages, means there is a pooling, and whenever you need, macrophage will come out and help you out in killing the foreign body. Okay, until called upon the sites of infectious object, and this call is basically in the form of some chemotactins that we have already discussed. So on the stimulus, they detach and become mobile macrophages. that respond to the chemotaxis. So the body has a widespread monocyte macrophage system in all the tissues of the body. So we will look what are the different tissues where these macrophages are present in the large quantity to give the defense to the body. But before the total combination, what is the monocyte macrophage system made up of? You can say it is made up of basically the monocytes, mobile macrophages, fixed tissue macrophages, and few specialized endothelial cells in the bone marrow, spleen, lymph nodes, all this collectively called the reticuloendothelial system. Because all these originate from the monocyte stem cells, reticuloendothelial system is synonymous with the monocyte macrophage system, and it is generalized phagocytic system located in all the tissues of the body. So remember, reticuloendothelial system is present in all the tissues of the body. And this picture is basically showing you that monocytes basically resides into the circulation in the normal condition where the permeability is very low or zero for the movement of monocyte to the outside. But once there is some chemotactin in the body, this chemotactin will cause increase the permeability between the cells, endothelial cells, so that these monocytes will squeeze through the interstitial space by the process of diapedesis and come towards the tissues and then convert into the macrophages. So basically, the components of reticuloendothelial system are primary lymphoid organs and secondary lymphoid organs. The primary lymphoid organs include all the organs where the cells of the reticuloendothelial system are formed and get matured, and this is bone marrow. So we have reticuloendothelial system, we have monocytes basically. So in the bone marrow there is a formation of monocytes. So the primary lymphoid organ is the bone marrow, and then we have secondary lymphoid organ. These are the basically the lymphoid organ. These secondary lymphoid organs are basically the sites where the cells of the reticuloendothelial system functions. Means as I told you that the macrophages are stored in tissues, and basically there are some sites over there on each and every tissue, so that the macrophages will be stored over there or attached over there. So the most common are the lymph nodes, tonsils, spleen, and MALT, mucosa associated lymphoid tissues, Kupffer cells, microglial cells. So these are different sites, or which are also known as lymphoid organs, secondary lymphoid organs, where the cells of the reticuloendothelial system will work. That is, they will be activated and start working as the foreign agent. So in the previous slide we have seen the MALT, mucosa associated lymphoid tissues. So MALT are of numerous types, like bronchus, MALT, bronchus associated gut, nose, larynx, skin, vascular, conjunctiva, which is related to eye. So we have different types of mucosa associated lymphoid tissues where the macrophages also work. So macrophages in the lymph nodes. Now we will discuss these lymph nodes, tonsils, spleen, one by one. Okay, so first of all, the macrophages in the lymph nodes. They are basically known as the checkpoints of the body, and all the particles, including microorganisms, after invasion of the tissues, do not enter directly into the blood. So first of all, they will enter into the lymph, and then they are trapped in the nodes in the sinusoids lined by the tissues macrophages. And then we have macrophages in the spleen and bone marrow. If the organism has been successfully past the lymph nodes, means lymph node was unable to stop the organism's foreign particles to enter, then it will enter the blood. But in the blood will pass, there's another obstacle, is the spleen. So the spleen is similar to the lymph nodes, except that blood instead of lymph node and lymph will flow. So the red pulp and the venous sinuses in the spleen are lined by macrophages where they are active in removing the invading organisms. Means initially there is entry towards the lymph, by the lymph into the and then there is entrapping, means the organism will be entrapped by the lymph nodes, but if it is not successful, these will enter into the spleen, and there is another macrophage system which is fresh, so that are active in removing the invading organisms as well as unwanted debris in the blood, like destroyed RBCs or remnants of the RBCs or other cells. So the another one is the spleen and the bone marrow, where another macrophage system is more. This is the swollen lymph nodes. You can see this is the system made up of. This is a lymphatic system. Now come to the tonsils. Four group of tonsils in the pharyngeal region provide effective defense mechanism against intruders trying to enter the body by the nose and the mouth. They all form Valier's tonsillar ring. I know everyone knows tonsils. So this is basically the Valier's tonsillar ring. Have got the same function as that of the lymph nodes, as they too have the macrophages lining their interior. So the tonsils are very important, and but I believe that this is the most important in prevention of entry of the foreign bodies. GALT is the gut associated lymphoid tissues, and GALT is made up of several types of lymphoid tissues that store immune cells such as T and B lymphocytes that carry out the attacks and defend against the pathogens. Pairs patches found in the small intestine, mostly in the ileum, are part of GALT, means GALT associated lymphoid tissues. So if you see the word pairs patches somewhere, so always remember they are present in the small intestine, and their role is basically serve as lymphoid tissues. And then we have macrophages in the skin and subcutaneous tissues. These are also known as histocytes. These macrophages are also known as histocytes, derived from circulating monocytes that have become stuck in the tissues and subcutaneous tissues, and they provide the first line of defense of the skin. This is associated with the skin. The skin get interrupted because of some damage to the epithelium. And then we have these macrophages system in the lungs, alveolar macrophages. These macrophages are present as the integral component of the alveolar walls. The fate or the destiny of the entering organism. If the particles are digestible, if the foreign body or the bacteria are digestible, then what will happen? Macrophages can also digest them and release digestive products into lymph. If the particle is not digestible, especially this is for the lungs, okay, if the particle or the bacteria is not digestible, its size is large, it has a protective covering, then what will macrophages do? It cannot be digested, so the macrophages form a giant cell, okay, giant cell capsule around the particle until such time, if ever, that it can be slowly dissolved. Means there is chances that it will not be dissolved, means it cannot be degraded into, or it is not defragment into small size, or maybe defragment, but very slowly. So till that time there will be this capsulation over there in the lungs. Then we have macrophages in the liver sinusoids, also known as Kupffer cells. If the organism entering through the GIT has been able to enter the GALT and submucosa of the intestinal tract, then it is carried all the way through the portal circulation to the sinusoids of the liver, where tissues macrophages are present. What does it mean? It means if it crosses the tonsils, if it crosses the GIT, GALT, and then enter into the circulation, still we have the chance to stop the movement of this bacteria by the portal circulation, because due to the portal circulation, this substance, this blood will reach to the liver, where the tissue macrophages are present, and it will effectively remove the invading organisms, thus none of them is able to enter the blood stream. Macrophages in the brain. Microglia of the central nervous system can be considered a part of reticuloendothelial system. Microglial cells are basically the supporting cells of the neurons. So they also give support and protection with the and serve as a reticuloendothelial system. So they are the scavenger or searcher cells that proliferate in response to CNS injury. So you see, almost all tissues have a huge macrophage pooling, you can say. Role of macrophages in inflammation. When injury occurs, multiple substances are released by injured tissue, which causes the secondary changes in the surrounding normal tissue. So the tissue changes are called inflame. So the inflame has some characteristics. Okay, so these characteristics are vasodilation, that area become vasodilated, increase vascular permeability for these macrophages, monocyte and neutrophil, sorry, increase vascular permeability, clotting of the fluid in the interstitial fluid, and migration of neutrophils and macrophages towards the target area, swelling of the tissues, and caused by histamine, bradykinin, serotonin, prostaglandin, lymphokines, and T cells, because these multiple substances are released by the injured tissues and they produce all these effects. These substances are activate the macrophages that eat and destroy the tissues, or the substances that are outside, there means dead cells or the bacteria. Then we have a walling off effect of inflame. Means one of the result of inflame is wall off the area of injury from surrounding tissues. Wall off means making a wall. Okay, so they will make a wall so that there will be chances that the tissue spaces in the inflamed area are blocked by fibrogen clot to delay the spread of bacteria or toxin. Tissue macrophages are the first line of defense against inflammation, because you know the tissue, as I told you, the tissue macrophages are the prepared macrophages, and whenever there is entry of a foreign particle, there is chemotactic substance, so there is a chemotaxis. So the tissue macrophages which are already mature, they will move towards their target and serve as a first line of defense against inflammation. And then we have a neutrophil, which are serve as second line of defense, and within 1 hour of inflame, the large number of neutrophil enter the inflamed area from the blood by migration, diapedesis, and chemotaxis and kill the bacteria. And sometimes the number of neutrophil increases 4 to 5 fold, caused by the product of inflammation. Means the area where is inflammation is increases, so there is release of some substances, some products like cytokines and tumor necrosis factor and other substances, so that will serve as the feedback, positive feedback, and they give signals to the bone marrow so that more and more neutrophils will be formed and released and come to the target point and destroy the foreign body. The second macrophage invasion of inflamed area, that is the third line of defense. What does it mean? Along with neutrophils, monocytes enter the inflamed area, okay, and enlarge to become macrophages. Means, first of all, already matured macrophages are over there, and then we have neutrophils which are present in the blood, and then the third step is the monocytes, because second macrophage invasion, because we need more and more macrophages. So what will happen? Monocytes will enter the inflamed area and enlarge to become the macrophages. This entry of monocytes and become enlarged is the third line of defense, because we are in because after the second line of defense we are in lack of macrophages, because already matured macrophages have gone to the inflamed area. Now we need more and more macrophages. So buildup of the macrophages in the inflamed area is very slow than the neutrophils and requires several hours to become effective. So the macrophages can phagocytize the far more bacteria and larger particles as compared to neutrophils. And finally, we have fourth line of defense. Increased production of monocytes and granulocytes by the bone marrow is the fourth line of defense. Means we have increased the production of macrophages. So of course we need the production of monocytes. Once the monocyte will increase, macrophages will be increased. So the fourth line of defense is the production of monocytes, and it take about 3 to 4 days by the bone marrow. If stimulate from inflamed area continues, means inflammation will produce more and more inflammatory products, inflammatory cells, inflammatory chemicals that give a stimulus to the bone marrow. So more and more monocytes will be produced, and the bone marrow continue to produce high number for months and years at the rate of 20 to 50 times normal. Means for example 50 times greater number of monocytes will be produced, for example in the case of cancer. And then we have feedback control of macrophage and neutrophil responses. So in this picture we can see this is the activated macrophage. Okay, consider that there is entry of any bacteria, and these macrophages are activated by some chemotactic, and then what will happen? This activated macrophage will release some substances. These are the cytokines, and these cytokines will act on the bone marrow directly, okay, directly, and increase the production of granulocytes, monocytes, and macrophages. The indirect route is the, first of all, these macrophages are activated, and then they release tumor necrosis factor and interleukin 1, which will act off endothelial cells, fibroblasts, and lymphocytes, and these cells will release further substances, and then they will act on community stimulating factor. These are basically the factors that stimulate the formation of blood cells. So they act on the bone marrow, and more granulocytes, monocytes, and macrophages will be produced. So in this way the whole monocyte macrophage system work along with the help of neutrophils. So they all work together. But basically the monocyte macrophage system is the conversion of monocyte to macrophages, and more monocytes will be produced, so that they will convert it to the macrophages, and macrophage will go and eat a lot of bacteria at a time, because it is of the large size. So this is about the basic mechanism of reticuloendothelial system. I hope you understand very well. At last I just want to say, जिसने रब के लिए झुकना सीख लिया वही इल्म वाला है, क्योंकि इल्म की पहचान आजिज़ी है और जाहिल की पहचान तकब्बुर में है. Thank you so much.